hello guys welcome back to another video. So today I'll be teaching you skeleton muscles, the working and the structure and the mechanism of skeleton muscles. So just to give you an idea of what exactly I'm teaching, in the NCRT you can see this diagram. So that chapter ka I will be teaching you skeleton muscles. So we will look into everything. We will first see the structure. Now I only plan to explain you the mechanism, but without explaining you the structure I won't be able to explain you mechanism very properly. So that's why I will first be explaining you to the whole structure. If you already know the structure and you are thorough with it, you can directly skip to the mechanism portion. But if you don't know anything about it, I will explain it to you from scratch. Now I'm assuming that there are certain muscles, and these are basic things which you might know, and which are locomotion, what is locomotion, all the definitions are given in the starting of the chapter. So I hope that you have read it all. Another thing I wanted to tell you, I will explain to you the mechanism and everything, but just after the mechanism there is a red muscle fiber and white muscle fiber. Those are just classifications. Those don't come on the structure or the uh, they do come on the structure, but they are a different type of structure. It doesn't come under the mechanism, so I will not be explaining to you that. It is just a simple reading part, so you can read that. And this is one of the most important topics. But another important topic from this chapter is the last page, in which the disorders are given. The disorders, one question every year comes. You please do the disorders thoroughly, and also do the joint part, the knee, ball joint, hinge joint, all the joint, they also come. So please do that thoroughly as well. So now without any delay we will start the structure first, then we will move on to mechanism. So skeleton muscles, I will be explaining you in the diagram. You can refer to it in your NCRT. So the skeleton muscles, first of all, they have number of muscle bundles or fascicles. So skeleton muscles, it is divided, then it is subdivided. मतलब skeleton muscles है, उसके अंदर muscle bundles है, और fascicles है. fascicles को fascia से cover करते हैं. उसको cover करने के बाद each fascicle contains a muscle fiber, और वो muscle fiber को further divide करते हैं. So I've made a proper chart. You can write this down and learn it as it is. So have number of skeleton muscles have number of muscle bundles or fascicles. Now the fascicles, very important question, the fascicles are covered by what? The fascicles are held by collagen connective tissue fascia. This is very confusing terms, please do it thoroughly. So the fascicles, these are held together by connective tissue called as fascia. Then each fascicles or each muscle bundle, it is further divided. Each has a number of muscle fibers. These are our main unit. So each fascicles में क्या है, number of muscle fibers है. मतलब you understand, like a body is there, a body has number of cells, many number of cells. So it's like that. So skeleton muscles have number of muscle bundle or fascicles. One fascicle has many muscle fibers. So the muscle fibers is now divided. The muscle fiber, it is lined by plasma membrane sacrolemma. मतलब उसका जो muscle fiber का जो plasma membrane है, that is sacrolemma. It is called as sacrolemma. Then sacrolemma covers the inside cytoplasm. It is called as cytoplasm. Okay, so what is this? वेरियड मसल फाइबर है. मसल फाइबर को कौन लाइन करते हैं, या फिर मसल फाइबर का प्लाज्मा मेम्ब्रेन कौन है? इट इज़ कॉल्ड एज सैक्रोप्लाज्म. नाउ इनसाइड द सैक्रोप्लाज्म, इनसाइड व्हाट इज इनसाइड प्लाज्मा मेम्ब्रेन? देयर इज cytoplasm, right? So the sacrolemma covers the inside cytoplasm, and it is in the sacromere, so it is called as sacroplasm. So sacroplasm, it is multinucleated, therefore it is syncytium. So जो भी multinucleated होता है हम उसको syncytium बोलते हैं. So it contains more number of or multiple number of nuclei, therefore it is called as syncytium. Another very important statement, sacroplasm is syncytium. Then ER, endoplasmic reticulum, that is the sacroplasmic reticulum, is the storehouse of calcium ions. Now very very important, देखो हर एक organelle, each organelle is given like its unique name because it is in the sacromere, right? Cytoplasm is called sacroplasm. The plasma membrane is called sacrolemma. Then the ER is called endoplasmic reticulum is called sacroplasmic reticulum. So very very another important, The ER, that is the sacroplasmic reticulum, is the storehouse of calcium ions. Very very important. It is the store of what? It is the storehouse of calcium ions, Ca++ ions. Another very important question. Now we will move further. I hope you understand the structure. You can take the help of diagram, but it is a bit confusing. So I have given you a chart. You can learn it according to this. Just a second. Okay. So now we will go further into the structure. Now this is the main important part. This was just the before part, like the classification and bit. This is the main core part. Please pay attention. The characteristic feature of muscle fibers are the parallelly arranged filament in sacroplasm called as myofibrils or myofilament. So what is the characteristics of muscle fiber? हमने देखा muscle fiber में क्या है, sacrolemma है, sacroplasm है, sacroplasmic reticulum है. The characteristic feature is that it has in the sacroplasm of the muscle fibers. Listen to what I'm saying. In the sacroplasm, not not sacroplasmic reticulum or sacrolemma. In the cytoplasm, that is the sacroplasm, there are parallelly arranged filament which are called as myofibrils or myofilament. These are very very important. Please pay attention. Parallelly, they are parallelly arranged. Each and every word I've written here, do it as it is. It will be very beneficial for you. Do it as it is. It has parallelly arranged filament in sacroplasm called as myofibrils or myofilament. So the myofibril, it has alternate light and dark band, rod like parallel. So अभी क्या होता है, see there is now it in further slide I will teach you. There is I band, there is A band, isotropic, anisotropic. Okay, we will do that in further class, but sorry, in further slide. But remember myofibril, you are understanding the flow. Where we were on skeleton muscle, right? Now we came down to the smallest unit, which is the myofibril. Now सारा जो खेल चलता है वो उस myofibril में चलता है, contraction and all that. So just you, if you copy this chart down, if you read it every time, you read it 5-6 times, it will sit in your memory. This is very very extremely easy. Like imagine I've just done my 12th and I'm able to explain you this. It is that easy. So please pay attention. So myofibril, it has alternate light and dark band. They are they are like parallel. And now I've drawn it this way to show that this is the dark band, light band, dark band, light band, dark band, light band. In physics you have done the Young's modulus of elasticity, sorry, YDS experiment, Young's double slit experiment. उसमें कैसे आते bands आते, right? Color के ऐसे light white. So just like that, it is arranged in bands. Okay, there is a dark band and there is a light band. This is the dark band, this is the, imagine that this is light band, this is the dark band. Okay, now we will further divide this into dark band is the what is dark band, what is I band. It is further divided. I will tell you that. Okay. So guys, so let's start with the detailed structure of myofibril now. Now I will teach you the light band, dark band and all of this. After that we will go into the detailed structure of actin, myosin, all that. So first let's learn this. So myofibril, it has two types of bands. As I told you in the earlier slide, it has the light band and it has the dark band. Now the light band, it contains a protein called as actin. We will go into the detail of what actin is, what is its structure, all that. But we have to look at it from a bigger point of view right now. So light band contains actin and dark band contains myosin. These are going to be your main proteins through which contraction happens. So please remember them. Light band contains actin and the dark band contains myosin. So now the light band, it is called as the I band or isotropic band. Now another name given to the light band, or when we learn contraction or when we learn the mechanism of contraction, at that time we will understand, we will refer to light band as the I band or isotropic band and the dark band as A band or an isotropic band. So isotropic means which emits light, right? So you can remember it like that. So this is isotropic I band, and dark band is called E band, which is anisotropic band. So actins are thin, therefore called as thin filament. So another important major important difference, the actin protein are thin. See, we are talking in comparison here, the myosin is thick and the actin is very thin as compared to myosin. So actin is called the thin filament and myosin is called the thick filament. Another, myosin are thick, therefore called as thick filament. You can take this chart down and learn it. So as we know that myosin are called thick filament and actin are the thin filament. I hope you understood this. Now let's get into the diagram which is given in NCERT. I will show it to you, you will recognize it, of course, because it's famous diagram, quite famous diagram. So this is basically the diagram. I hope you can see it clearly. This is the diagram of sacromere. Now I have personally drawn it and labeled it in the diagram itself what every component means. So please, if you can take a screenshot of this or you can draw it yourself, but please do it this way. If you go statement wise and you do diagram separately, it will be very confusing for you, but if you do it like this, it will be much easier for you to remember. So now I will just explain it to you everything. So these are basically, so we will basically start with what we have learned, the I band and the E band. As we know, this is the structure of the sacromere. So this is the I band. As we talked, I have made them thin. They are the thin I band, and this is the A band. Okay, this is the I band, and this is the A band, the thick one. Okay, I band, and this is the A band, the thick one. Now there are certain terminologies used in a sacromere to define where exactly is the sacromere. So there are two Z lines. So this line and this line, these are called the Z lines. So इनके बीच का जो area होता है, the look, what is Z line क्या है? Sacromere is between two Z lines. You will learn everything, but follow me as I'm teaching you. The area between two Z lines, this is the first Z line, this is the second Z line. Area between two Z lines is called sacromere. What is sacromere? Sacromere is the functional unit of contraction. मतलब when mechanism of contraction occurs, everything happens in the sacromere. It shrinks, it relaxes and all that. It happens in sacromere, so it is the functional unit of contraction. So again I'll repeat myself, sacromere is the area between two Z lines. Okay, so that is done. So now we will move that what another term, Z line is the area, two Z lines के बीच में जो area होता है उसको sacromere बोलते हैं. Another thing to remember about Z lines, it bisects the A band or thin filament that attach firmly. Now I have not drawn it here, like इधर ऐसे further ऐसे होता है, मतलब ऐसे further जाता है और ऐसे continue होता है, ऐसे Z lines ऐसे चलता जाता है. तो ये जो अभी इधर you are seeing here is there is one I band here, there is one I band here. So basically a full I band is there, and this Z line, this is bisecting it. Okay, it is dividing into half. So that's why we call it that it bisects I band or the thin filament. What is I band? I band is also thin filament, right? So thin filament attaches to it firmly or it bisects the I band. मतलब what are you seeing here? This is the Z line, right? This is the Z line and इस पे attach, right? I band इस पे attach है. So that is one definition. Thin filament attaches to the Z line firmly or that it bisects the I band. मतलब इधर भी जा रहा है और इधर भी जा रहा है, दोनों के बीच को bisect करता है. Now there is another line which bisects the A band, which is the anisotropic band or the dark band. That is called the M line. M line. It bisects. I will return here. This is very important diagram. It bisects the A band or thick filament or holds together the thick filament. So this is the M line. So इधर कैसे you can see that it is now in the NCERT diagram. It is not given. It is only mentioned in the text. I have drawn it over here for you. So it holds the it holds the A band together. So M line and Z line are too similar. Okay. Z line क्या करता है? It bisects the I band and it holds together the actin filaments or the I bands. But in here M line, it divides the dark band or the thick filament and it attaches the thick filament together. Okay. So learn the M line, learn the Z line. उसके बाद what is H zone? So as you can see in the diagram, this is the I band, this is the A band. I band and A band, they are overlapped. You can see, right? This area belongs to both I band and A band. This area, they are overlapping over each other, right? But here you can see, is it overlapping? Here is there somewhere? There is no I band right over here. So the area in which it is not overlapping, the thick filament is not overlapped by the thick filament. This thick filament is not overlapped by the thin filament, is called as the H zone. I have written it here. Central part of the thick filament not overlapped by the thin filament. These parts are overlapped by the thin filament, but this central portion, it is not overlapped by the thin filament. So the portion of thick filament which is not overlapped by thin filament, this is called as H zone. Okay, so I guess I've covered everything. I hope you understand this diagram of sacromere. Now further we will go into detail of actin and myosin. They have further structures, and then after we do those two structures, then we will start with the mechanism. Okay. Sorry guys, in the previous slide my voice was cut, but I explained to you the structure, the sacromere ka structure. Now if you really want the voice, now I've the way my slide was, I've written everything down, so I was just explaining those points to you, and I explained here and there a few points. But if you listen to it closely, you can hear them if you have earphones or something like that. But if you want me to make another video regarding that, I can make it. But really, if you do the notes, I think you'll be fine. And I'm extremely sorry, my earphones were connected, but they were not connected properly, so the sound did not capture in the microphone. I will take care next time. But if you want a video on specifically that, if you can't understand and still want me to explain it to you, I can do that. But if you do the diagram properly and if you learn what is given in the diagram, you will be fine. So now let's start with the individual प्रोटीन, द एक्टिन एंड द मायोसिन. इंडिविजुअली हम इनको पढ़ेंगे, उसके बाद हम मैकेनिज्म पे जाएंगे. सो एक्टिन, so it is also subdivided how it is made and what is made, and there is a diagram of it given in NCERT. So now I'll start. So actin is made of two filamentous actins helically wound. मतलब एक्टिन एक प्रोटीन है, वो किससे बना है? 2F एक्टिन. एक्टिन एक होल प्रोटीन है और F एक्टिन उसका सब यूनिट है, तो 2F एक्टिन मिला के एक एक्टिन बनाते हैं. सो ये 2F एक्टिन, दीज आर हेलिकली वाउंड टुगेदर. These are called as filamentous actins, means two filamentous actins form one main actin, and the F actin is a polymer of monomeric globular actin. मतलब कैसे में एक मेन एक्टिन है, एक मेन एक्टिन किससे बना है? दो फिलामेंटियस एक्टिन से. और ये जो फिलामेंटियस एक्टिन है ये एक पॉलीमर है, तो पॉलीमर किससे बनता है? मेनी मोनोमर्स फॉर्म वन पॉलीमर, राइट. सो दीज मोनोमर्स जिससे फॉर्म होता है उसको बोलते हैं हम ग्लोबुलर एक्टिन. मतलब समझ 10 globular actin collect together and form one filamentous actin. Then like this, two filamentous actins form one actin. Now globular actin का number नहीं दिया हुआ है, but a very important filamentous actin का दिया हुआ है. How many? Two filamentous actins form one main actin, and many globular actins or G actins form the F actin. Okay, so we are clear with this. You can learn this in a chart. Please learn it in a chart. So two filaments of another protein run close to the F actin. These are called as tropomyosin. So जो ये अलग है. So जो F actin हमने पढ़ा, जो एक्टिन मेन बनता है, F actin है. F actin ऐसे गोल गोल होता है और उसके साइड से कि ये जाता है, जैसे DNA कैसे होता है, मतलब हिस्टोन प्रोटीन कैसे होता है. हिस्टोन प्रोटीन एक गोला होता है और DNA उसके अराउंड व्रैप होता है. वैसे यहाँ पे जो वो गोला है वो है हमारा एक्टिन, और जो व्रैप अराउंड हो रहा है वो है हमारा ट्रोपोमायोसिन. एंड ऑन दिस ट्रोपोमायोसिन ऑन रेगुलर intervals another protein called as troponin is present. Now I will explain to you through a diagram so you will understand it better. You can please note it down or take a screenshot of this. By the way, in the previous slide you can also pause that video and then you can take down the notes. and learn it notes wise. I think that is more valuable than to listen to me, I guess. Notes, 1 second. So this is the diagram. I hope it's visible. So this is the diagram of actin, which is the thin filament. So these गोल you are seeing, these are the F actins, and उसके along क्या run करता है, ट्रोपोमायोसिन, जो ग्रीन कलर से दिखाया हुआ है, जो ग्रीन thing, this is the tropomyosin. These are another pigment, and on this tropomyosin, not wholly, on regular intervals, are another protein called as troponin present. And what's important, this troponin are the main thing in our contraction ka process. So troponin, it forms the active binding site for myosin. When we learn the structure of myosin, I will tell you what this statement exactly means. But now remember that the troponin, it forms the active binding site for myosin. Please draw this structure or learn it, or take a screenshot and learn it. Very, very important. Now we will go on and learn the structure of myosin. Okay guys, so let's start with myosin. So we just did actin, now it's time to do myosin. So myosin, as we know, it is the thick filament. So thick filament, it is just like actin, it also has its structure and subunits. So it is a polymeric protein formed by many monomeric protein. These are called as meromyosins. So many meromyosins form the myosin protein. So meromyosin, it is a monomer. So many meromyosins collect together and it forms a polymer, which is the myosin. Okay, so the meromyosin. Now further, meromyosin is further divided into globular head and short arm and a tail. So it is divided into two parts, not three parts, two parts. Globular head plus short arm, they come collectively. Globular head plus short arm is one part and tail is another part. So it is subdivided into two parts, the globular head and short arm and the tail. The globular head and short arm, it is called as the heavy meromyosin. So it is the larger portion or the bulkier portion. So it is called as the heavy meromyosin, or the heavy part of the meromyosin. And the tail, it is the lighter portion. So it is called as the light meromyosin, or LMM or HMM, ऐसा हम बोलते हैं. So in the diagram I will explain it to you more clearly. So this heavy meromyosin, or the globular head and short arm, it project outwards and form a cross arm with actin. So when contraction happens, there I'll teach you how this cross arm is formed. But when sliding or contraction happens, this heavy, that globular head and short arm, it bulges out. It projects outward and it forms a arm, or like a structure with the actin. It is called as a cross arm. This is formed by which part of the meromyosin? The heavy meromyosin. This is very important. Learn this whole structure as well. So now I will explain it to you in the diagram. Guys, I won't say that it takes a lot of efforts to make this. It does, but it's none of your concern. You're here to learn. So if you like what I make, and you like how I teach, or the notes or the diagrams which I give you, or the overall chart purposes, how I teach you, please like the video. And you don't need to like worry about how much I'm taking efforts to make this. You're not telling me, I am doing it for you. So if you would subscribe, it will be really good. So let's get back to the diagram. So as we know, myosin का हम diagram कर रहे हैं, thick filament. So this is the diagram given in NCERT. This is the tail portion, or the LMM, light meromyosin. This is the head portion, or the HMM, heavy meromyosin. So ये है ग्लोबुलर हेड, और ये क्रॉस आर्म फॉर्म करता है. दिस इज़ द क्रॉस आर्म. नाउ वेरी वेरी इंपॉर्टेंट थिंग, सीधा डायग्राम क्वेश्चन में आएगा. और डायग्राम में कैसे है, एनसीईआरटी में दोनों ब्लैक ब्लैक दिया हुआ है. सो दी ऊपर वाला, ऐसे दे विल गिव यू टू होल्स, एंड उसमें से यू हैव टू रिकॉग्नाइज विच इज़ विच. सो दी अपर वन इज़ द एक्टिन बाइंडिंग साइट, I will explain it to you. What is the actin binding site? Why it happens? I will explain it to you. But just now focus on the diagram and learn this. So the upper one is the actin binding site, and the lower one is the ATP binding site. This will come in your coaching tests or whatever. In this diagram, the upper one is the actin binding site, and the lower one is the ATP binding site. So I hope you understood the diagram of myosin. Now we will move to the actual mechanism of contraction and learn what it has to teach us. Okay guys, so let's now start with mechanism. So I'm shooting this video on next day because yesterday the light went off, but anyway, let's continue. So now we have learned the structure of actin and myosin. So now we will move on to the mechanism of muscle contraction. This is the main part which you are waiting for. So let's start. So it is a mechanism of muscle contraction is best explained by the sliding filament theory. So this theory, we in the NCERT, no name is given by whom this theory was given, but it can be asked which explain the best mechanism of contraction. So it is the sliding filament theory. So what does this sliding filament theory? It states that the contraction of muscle fiber takes place by sliding of the thin filament over thick filament. Means जो actin है thin filament, it slides upon myosin, which is the thick filament. Very very important. It is sliding of thin filament over thick filament. They can give ऐसे उल्टा पुल्टा करके statements, but you have to remember this clearly, that it is just there is sliding of thin filament over thick filament. Okay, now another important thing to learn, the junction between motor neuron and sacrolemma is called neuromuscular junction or motor end plate. आगे when we see the sequence of how the muscle contraction occur, there will be one neuromuscular junction. So I just wanted to clear it out where that neuromuscular junction is present. So this junction is formed between motor neuron and sacrolemma. Very very important, learn this. Motor neuron and sacrolemma के बीच में एक junction होता है, that is called as neuromuscular junction or motor end plate. Now we will start with the mechanism of contraction. Okay, now let's start with the mechanism of contraction. So these are the steps given. I will read it to you one by one and then explain it to you. So for contraction, first a signal is sent by CNS. A signal is sent by CNS via motor neuron. मतलब जो मोटर न्यूरॉन जहाँ पे तुमको कॉन्ट्रैक्शन या फिर जो भी करना है, तो उसके लिए जो CNS है, दैट सेंड्स अ सिग्नल टू दी VI, मतलब बाय मोटर न्यूरॉन. मोटर न्यूरॉन के थ्रू CNS सेंड्स अ सिग्नल. नाउ वेयर डज इट सेंड द सिग्नल? न्यूरल सिग्नल reaches the neuromuscular junction. As we read the junction between motor neuron and sacrolemma, that is called the neuromuscular junction or motor end plate. So that उधर जाता है signal. So the signal reaches to the neuromuscular junction. So from then, what does neuromuscular junction now do? The neuromuscular junction now releases neurotransmitter acetylcholine. What neurotransmitter? Acetylcholine, which generates action potential in sacrolemma. Now what happens when this signal reaches neuromuscular junction? Neuromuscular junction releases certain neurotransmitters which are acetylcholine, which generate an action potential in the sacrolemma. Okay, these are the steps, please learn it as it is. So an action potential is developed in the sacrolemma. Now what does this action potential do? So this action potential causes the release of Ca plus ions. What ions? These causes the release of Ca plus ions in sacrolemma. Okay, यहाँ तक clear है? 4 steps clear है? Learn these 4 steps. Now we will move on to the 5th step in the next slide. But remember, importantly, हमने पढ़ा था what is where number of in the sacroplasm reticulum it has large number of Ca plus ions. I read it to you in previous slides. So this causes a release. उधर से क्या होता है? It causes a release of Ca plus ions in sacrolemma. मतलब जो sacroplasmic reticulum है उस उधर से Ca plus ions release होते हैं. कहाँ release होते हैं? Sacrolemma में. Due to what? Due to action potential generated by neurotransmitter acetylcholine. Now we will move on to the 5th step. Okay, guys. So now the sixth step, or the fifth step. So what happens, as we know that CA plus sacrolemma में collect हो जाते हैं. Now very important. This fifth point is very important. So the CA plus amount increases. That is, the CA binds with troponin on actin filament and remove the masking of active sites, therefore making them available for myosin to bind. Wait, I'll explain it to you. So what happens on troponin? What happens? The CA plus sacrolemma में आते हैं, and what they do, they go and attach to the troponin. We read in the structure, actin have small proteins called as troponin. So what happens? The CA goes and it attaches on troponin, and it removes their masking site. Now what is masking site? What happens in contraction, right? So the myosin filament and actin filament, they get joined and then they form a cross bridge. In order for that cross bridge to happen, they should attach. So for that attachment, it's like हमने कहीं पे ग्लू लगाया हुआ है और उस पर हमने कवरिंग बिठा दिया है. तो अभी हम कुछ भी उस पर चिपकाएंगे तो चिपकाएगा नहीं ना, क्योंकि उस पर कवरिंग है. तो वी हैव टू रिमूव दैट कवरिंग एंड देन वी कैन प्रेस एनीथिंग एंड वो चिपक जाएगा. इसकी वजह से हमको वो कवरिंग हटानी है. सो व्हाट इज़ दैट कवरिंग? So the removal of that covering is called removing its masking. So what does this Ca plus ion do? It binds with troponin on actin filament and removes the masking of active sites. मतलब जहाँ पे मायोसिन को जाके अटैच होना है वहाँ की ये मास्किंग साइट हटाता है ताकि मायोसिन जाके अटैच हो सके. अगर Ca+ होता तो अटैच नहीं हो पाता. ओके सॉरी, अगर Ca+ नहीं होता तो अटैच नहीं हो पाता क्योंकि मास्किंग वहाँ पे रहती है. So it removes the masking so that myosin and go and attach, making them available for myosin to bind. This is only written, so please learn this. Now what happens? The myosin uses ATP and binds to actin to form a cross bridge. Now myosin ऐसे ही तो attach नहीं होगा. It takes help of ATP. हमने देखा था ना, ATP बाइंडिंग साइड भी होती है और एक्टिन बाइंडिंग साइड भी होती है मायोसिन के स्ट्रक्चर पे. तो it uses ATP and binds to actin, and what does it forms? It forms a cross bridge. There is cross bridge formation. Okay, so cross bridge formation happens. This pulls the actin filament I band to the center of A band. Now this is why the structure is very important. As you have learned, ये वाला structure था ना, ये ऐसे दो Z lines थे, ये एक्टिन था, ये बीच में ये ऐसा फिलामेंट था, फिर से ये था और ऐसे था. So this was your myosin, this was your actin, these were your Z lines. So what happens? The actin filament gets pulled towards the center. मतलब ये जो एक्टिन है ये ऐसे सेंटर की तरफ पुल हो जाता है इस डायरेक्शन में, और ये वाला इस डायरेक्शन में ऐसे ऐसे सेंटर की तरफ पुल हो जाता है. सो बिकॉज ऑफ एन एक्टिन इज अटैच टू विच इट इज़ अटैच टू Z line, right? It is attached to Z line. So Z line gets pulled inside because it's attached to actin I band. So Z line भी inside में pull होता है क्योंकि ऐसे ऐसे पुल होता है क्योंकि इट इज़ अटैच टू एक्टिन. सो एक्टिन पुल होता है तो वो साथ में Z line को भी पुल कर देता है. एंड बिकॉज ऑफ दिस देर इज़ अ शॉर्टनिंग ऑफ सैक्रोमियर. सो इससे क्या होता है, हमने क्या पढ़ा था, ये जो रीजन है ये सैक्रोमियर है. तो अगर ये दोनों अंदर आएंगे तो देर विल बी शॉर्टनिंग ऑफ सैक्रोमियर. सो शॉर्टनिंग ऑफ सैक्रोमियर से क्या होगा, कॉन्ट्रैक्शन होगा, एंड दैट कॉजेस योर कॉन्ट्रैक्शन. सो वॉट एग्जैक्टली डिड वी लर्न? That troponin comes, it removes the masking so myosin can bind. So because of this the I band, remember not the A band, the I band, these इनका कुछ रोल नहीं है. आई बैंड है, वो आई बैंड कॉन्ट्रैक्ट होते हैं या फिर पुल होते हैं अंदर की तरफ इन द मिडल ऑफ द ए बैंड. सो वी विल नाउ लर्न दैट व्हाट गेट्स रिड्यूस्ड एंड व्हाट ऑल. आई विल गिव यू स्लाइड ऑन दैट, बट प्लीज रिमेंबर, अंडरस्टैंड द कंसेप्ट हियर. सो द आई बैंड इज गेटिंग reduced ऐसे reduce and that is attached to Z line, so it pulls Z line as well inside. So because of that a sacromere, which is the unit of contraction, that also gets shortened, and because of that contraction occur. So this was your whole mechanism of contraction. I hope that in each slide I have given the steps. You can write it down on one big page or however in steps as you like it. So now we will move further, and I will give you a further more detail about relaxation as well. Okay guys, so we have completed contraction. So now these are two important points, so let's just read them. The myosin, when the contraction is done, at that time this process is occurring. So the myosin releases EDP plus PI, which is phosphorus, and it goes back to relaxed state. Therefore the cross bridge is broken. When contraction occur again, new protein comes and process continues. इसमें क्या बोला है, मतलब in your NCRT diagram you can see that there is a round diagram given in which a cross bridge is formed and it is again broken. So this process continues, and what happens when it is about to break, myosin releases, it is very very important, please learn this, the myosin releases EDP and phosphorus 1, and then the cross bridge is broken and the myosin comes back to relaxed state, and इसके बाद again a new protein comes and again the same process is repeated. Now we will see till when is it repeated. The process continues till all the Ca plus ions are pumped back into the sacroplasm cisternae or cisternae. So till when this process of breaking and again new protein comes or the contraction occur, it occurs till all the Ca plus ions are again sent back to the sacroplasm cisternae. So therefore, and why is it why does it continue till then? Because when all the Ca plus are sent back to sacroplasmic cisternae, then it making actin masked again. So what happens, why was contraction taking place in the first place? Because masking हो रहा था, right? Masking was removed by Ca plus ions. So if you send these Ca plus ions back from where they came from, then और अब masking कौन निकालेगा? So the masking cannot be removed. So the actin का masking is gained back. So actin is masked again. Therefore contraction stops. So Z line comes back to their position. Sacromere length restored. Therefore relaxation. So you understand, this is pretty easy. मतलब why was contraction happening? Because Z line was getting reduced, Z line was coming closer, sacromere length was getting decreased, therefore contraction was occur. So now the masking has come back again, myosin cannot bind, therefore length is restored, the Z line are back to the original position, therefore causing relaxation. So now we will see द कॉन्ट्रैक्शन में जो हमने डायग्राम किया था, जिसमें एच जोन, जेड लाइन, एम लाइन, वो सब थे. तो ये अगेन एक इम्पॉर्टेंट है कि ड्यूरिंग कॉन्ट्रैक्शन, दिस इज स्ट्रिक्टली ड्यूरिंग कॉन्ट्रैक्शन, what all is getting reduced and what. So the I band length decreases. We know this, right? I band length का decrease हो रहा है, I band length decreases, edge zone is also decreasing. So as you remember, edge zone is the part in the center where actin does not overlap myosin. मतलब myosin अकेला होता है, actin overlap नहीं करता है. पर अगर actin अंदर आएगा तो वो जो overlap ना करने का जो एरिया है, मतलब सिर्फ myosin वाला जो एरिया है वो डिक्रीज होगा ना, क्योंकि अभी जैसे इनके बीच का जो एरिया है, सो सॉरी, इनके जो बीच का जो एरिया है, मतलब सी दिस, ये जो एरिया है, तो ये कम होते जाएगा, राइट? एक्टिन कम्स क्लोज, सो दिस रिड्यूसेस. सो इसकी वजह से एज जोन क्या होता है, एज जोन डिक्रीजेस. द सैक्रोमियर लेंथ ऑल्सो डिक्रीजेस. हमको पता है, ये हमने देखा है. अभी जेड लाइन भी पास में आते तो सैक्रोमियर लेंथ इज डिक्रीजेस. द डिस्टेंस बिटवीन टू जेड लाइन्स डिक्रीजेस. Though two Z lines come closer, therefore their distance decreases, they come closer. Now these 4 points are very important. सीधा statement, मतलब choose the following में ऐसा आएगा. During contraction what all processes occur? So you have to tick, yeah, this occurs. फिर दे देंगे कि edge zone increases, edge zone does not increases, it decreases, so वो false हो जाएगा. So another this I have written this in red, okay. Sorry. A band, I have written this in red, this is a very important point. So the A band is unaffected. We have written that we have learned that I band decreases, edge zone, this, that, but very very important, during contraction A band is unaffected. Please learn this, A band is unaffected. Now another important point, the A band is only affected, only holds your 4 marks. This only basically holds your 4 marks. It is only affected during maximum contraction. नाउ चेक, ओपन योर एनसीईआरटी, उसमें दिया हुआ है रिलैक्स स्टेट, कॉन्ट्रैक्शन एंड मैक्सिमम कॉन्ट्रैक्शन. नाउ प्लीज़ लर्न दोज डायग्राम्स, मतलब गो थ्रू दोज डायग्राम्स. उसमें जो मैक्सिमम कॉन्ट्रैक्शन वाली स्टेट है, ए बैंड सिर्फ और सिर्फ उसी में अफेक्ट होता है, बाकी कंडीशंस में ए बैंड इज़ नॉट अफेक्टेड. ओके, so this is another important point. Now we are pretty much done with everything. I will just tell you about those diagrams. Check out the diagram in which cross bridge is formed, and then it is pretty easy, you can see from that. And check out the maximum and minimum matlab maximum and relaxed state ka contraction. And at the back of the textbook, as I told you in the start of the video, that check out the disorders and check out the joints ka part. One question guaranteed comes. So that was it about contraction, structure and mechanism of contraction. So if you like the video, please press that like button and consider to subscribe if you can. It will really motivate me to make more videos for you such like that. And please write in down in the comment section, what further videos do you want about in biology. And as of now, this is my 4th or 5th video, and I have only 1 subscriber, so I don't know if that person is even real, to be honest. But if you are real, whoever it is, please comment down. And yeah, thank you for watching. I hope you have gained some knowledge, and best of luck for your NEET, and do well. Thank you.